Okay, so nice to meet you all. I'm Florian. I'm coming from France, so a bit of a long travel to try to convince you that the beta distribution is really great. So that's what I'm going to try to do. And that's mostly a work I've been developing with Jason Wolf when I was in the UK. So to do this, I need to introduce you to the kind of model that is known to use this distribution. It's called as the Baldwin-Nichols model, but I added Ranelagh and Hartigan because basically they developed the same model at the same time. And like if you use the anagram, you can basically rewrite it in a different way to express everything it gathers in a sense. And you can say it's a kind of beta random herding null model. When I asked ChatGPT if herding can be used in that kind of sense, and it said no. But I still used it because I only listen to it when it agrees with me, which is helpful. So the basic thing you need to start with is like some kind of Moran-like model. You have a source population with a lot of individuals. Like, let's say they have beards. I will come back on this later. That's not my fault. And you draw an individual because it's going to migrate to form a pair. And then, like, once you have this individual in the pair, obviously it can reproduce at a rate B. And there's another way to build that kind of pair. Like the other way would be you draw a first one through migration, and then there's a second migration event. And this goes back to the classical view of population structure in traditional population genetics. Like you can basically compute the probability of having each of these pairs. It would be M times B or M times M. And then you can go back to the probability of being identical by descent, so F, and it would be 1 divided by 1 plus theta, and theta can be related to M and B, basically. So obviously, then, like, the thing that you want to ask is, can I build larger groups? Because, like, pairs are not very interesting. I mean, I don't know for you. For me, they're not very interesting. But you want to draw trios, and in these trios, like what you have is now you can have birth by the first individual, or you can have birth by the second individual. So you have this kind of reinforcement process. But migration is a kind of constant process. So you always have this kind of M from the migration rate, and now you have 2B. And the very cool idea that all these authors had is that you can relate this process to something called a Polya urn. And this kind of urn works in precisely the same way. So basically, let's say you start with a source population that is of size M. Like you draw an individual from that urn, and then rather than just adding back this individual in the population, you add an extra B individual to that source population. This is where this kind of reinforcement process comes. And then you can start all over again and draw a new one. And that's how you're going to build your trios. And the thing we know is that this Polya urn scheme is exactly describing what leads to a beta-binomial distribution. And this means that then you can add like multiple alleles in the population. You can have green beards, black beards, and this will follow this kind of beta-binomial distribution. Just need to know the population structure, so the kind of M over B, and the respective probabilities of if each of the genes or the individuals. And obviously, you can also extend this framework to more alleles. The infinite case, I think, gives the kind of even sampling formula. And so you have all these alleles, and it's going to lead to a Dirichlet multinomial distribution. And this model, to me, is some kind of null aggregation model. So that's where the herding part comes, because it's kind of saying the reinforcement process is purely linear. Like it's B, then 2B if you have two individuals, 3B if you have three individuals. So that's purely linear. There is no synergistic effect in the aggregative process. And it's also entirely independent from the number of alleles that you have in the population. Like if you drop the blue beard here, you have the same F in the population. If you have 10 alleles, you match one allele against the rest of the population, it's exactly as having one allele against a second allele. And the last thing is this kind of structure, it can represent basically any cause of non-random self-assortment. So that's the phi parameter that I define here. So that can be identity by descent, but that can also be in other sense, some kind of affinity that would bring more individuals at a rate that correspond to the B. And that would be another way to see it. Now, just to explain why we got interested into that distribution, I mean, that's Jason who found it at first because he has incredible skills at Googling stuff. Like he would find everything that is possible. And so we were interested into altruism, like very old question, many answers to it. We hope that they are kind of similar in what they mean. Yeah, that's the hope. So altruism, what it is, like altruism happens when you have an altruist individual that is going to pay a cost C to provide a benefit B. So here the benefit is a comb because maybe individuals need to brush their beard. I don't know, like, but yeah, I needed a picture for it. And the thing with altruism is that it can go to basically any individual in the population. So this means that if you look at the fitness of the different genotypes, you basically have the altruist who's paying the cost and having a bit of the benefit, and you have the cheater that just has the benefit. That's really something called as the social dilemma. Why the hell would I pay a cost to help other individuals? Like, that doesn't really make sense. And the way it was kind of solved, or at least one way, was to say, this should be solved if, in a sense, the altruist genotype finds a way to interact more often with itself. And that's the thing that is described by the self-assortment parameter here in the fitness, the phi. And in this case, like you just end up with Hamilton's rule, so very famous, very controversial. I'm not going to enter into those details. I added a phi here rather than the R because like I don't think it's only relatedness, and I think that also part of the debate was about this. So for me, it's just going to be self-assortment, and it's kind of the relatedness in Hamilton, like because in a sense, it's just how much more often I see myself than the other ones when I interact. So this kind of PA experienced by A minus PA experienced by C. Now to explain why, in a sense, because that gives an explanation, but why we were dissatisfied partly with that problem, I need to explain our system, and our system is social amoeba. And so social amoeba has this kind of amazing life cycle, I mean, it's amazing to me at least, where like all the cells are living a solitary life, and upon starvation, they're going to aggregate, and they will form this kind of almost animal-like, like it's a slug, it's going to move, it's dirty, I guess. I mean, I don't know. Like, but then it's going to form this kind of fungus, like a fruiting body and the cap where all the cells are going to reproduce. And the thing is, the cells in the cap are going to disperse and to form the next generation, but the ones in the stalk are going to die. That's cell sacrifice. And the thing that you observe when you really look carefully at them is that they're doing some kind of facultative cheating, which means that if you have different genotypes in the population, like when there's a rare one that interacts, it's always going in the cap. So it's cheating on the other ones because it knows that the other one is going to cooperate. So why would I bother? Why would I pay the cost? And so this led us to try to understand that question and to go to green beards, so that where the beards come from. And I'm going to focus on the kind of juicy things, to me at least, again. So I will assume that altruism has already evolved, like Hamilton's rule is satisfied. So to start with, I need to define what a green beard is. And I mean, I don't know why there's a beard. Hamilton had no beard, so I don't know if he was trying to solve something about himself. But like you have a green beard genotype, and it's only going to cooperate with itself. So when it meets itself, it's cooperating. Now you have an unconditional altruist, no beard color is going to cooperate with itself, but it's also going to cooperate with the green beard, which means that the green beards are cheating on altruists. What this means in terms of cooperative pattern is that unconditional altruists are always cooperating. That's, I mean, the definition. But greenbeards are only cooperating in parts of the group and mostly when they are frequent in the group. Otherwise, they're going to cheat because, in a sense, it's better. Like what this implies then is that the best fitness that altruists should have is if everyone is cooperating. But that's not the case when greenbeards enter into the population. And what greenbeards do is that they cheat on altruists when it's best to cheat, and they cooperate when it's best to cooperate from their perspective. And in all the groups, they are going to have a higher fitness. So what this implies is they're going to invade in the population. There's no way it could be, I mean, in a different way. So they're going to replace the unconditional altruists. And then this raises a new question, which is like, what is going to happen if you have a new beard color in the population? So now you have a blue beard in the population. And like the blue beard is going to cooperate with itself, but not with the green beard, and same for the green beard. So they behave according to their tag. And now you introduce a blue beard in the population, and you end up with something known as Crozier's paradox, because this blue beard is lonely in the population. So it's going to knock on the door of everyone begging for cooperation, but there will be no cooperation from the other ones because there is no one of its kind in a sense. And what this means is that the blue beard is going to disappear from the population, it's driven out, and you end up with basically, again, only green beards. And if there's only one color of beard, how can you discriminate the other ones? You can't really. So that's unconditional altruism. And this is something kind of related to what was known as tides of tolerance or intolerance, depending on whether you're optimistic or not. And what this means is you're kind of stuck between an altruist population and a green beard population, and you go from one or the other, but that's the only thing you can do. Like hyperpolymorphic green beards would be some kind of pie in the sky, in a sense. I mean, coming from Britain, sometimes I prefer the pie to be in the sky than in your plate. But I mean, that's personal opinion. Hope that there are no British people in the room. Like the thing I wanted to, so I wanted you to tell then is like, are we missing something? Is there more than meet the eyes here? And we do think there is, because like it has been focusing all these frameworks on pairwise interactions where basically you have groups of two and they do not cooperate with each other. But what happens if you have larger groups, like groups of three, for instance? Like maybe the green beard is going to cooperate when it has two copies of itself, because it's more present in this group than the blue beard. And well, that's where the beta binomial and the beta distributions were really useful, because we needed to account for the distribution of groups, not just the distribution of pairs, and not averages of these things. So we introduced this kind of population of genotypes, and now we draw groups in this population following this beta binomial distribution with this kind of theta parameter that is linked to assortment, so the phi parameter. And what we saw is that then there is no longer Crozier's paradox. So greenbeard polymorphism is going to evolve like almost every time you have a larger group, like G equals 3 is enough. And it's just because, as I was explaining, if you have large groups and a kind of public good system, like the rare bluebeard can kind of free ride in the groups when it's rare. Like the other ones are cooperating, I don't cooperate, I still reap the benefits in a sense. And the last thing that we saw is that like the more highly beneficial cooperation was and the lower the assortment was, the more you would see some kind of hyperpolymorphism. So we kind of think that social amoeba, for instance, would be in that kind of not so high assortment level and quite highly beneficial cooperation. Now, I told you that like the beta distribution had kind of become my obsession lately. And I mean, I want really to convince you that it's important, because I'm seeing it everywhere, even if it's nowhere. That's the problem maybe. But like this beta distribution, to me, it could also be some kind of null model of community assembly. Like you say, for instance, you take a source population with two strains. They are totally unrelated. And like maybe it's what the food provides you. Then like if you have some kind of migration reproduction process, like in the gut, for instance, maybe this kind of related weight between migration and reproduction can explain the variance that you obtain when you build different communities. So that's my kind of first idea to extend this framework. And the second one is looking at a more microscopic scale, because that's where I come from. And it's looking at compartments in the cell and what happens in metabolic networks. And obviously, like if you have like a proper compartment with an envelope, like every processes related to that are going to be stuck in that compartment. So that would be some kind of perfect self-assortment, like phi equals 1 in a sense. But like there's something called substrate channeling, and to explain it, I need to explain what it's not. Like what it's not is the kind of classical view of metabolism. You have a substrate, a first enzyme that is going to lead to a first product, and this first product can then be converted into a second product by a new enzyme, a different one. What metabolomes do is that they channel the substrate and the product by aggregating multiple enzymes together. And in this way, you avoid the kind of diffusion process in the cell. Like everything is kind of packed in these metabolomes. And it's still leaky. It's not as perfect as, so it's known as membraneless compartment. So it's more leaky than obviously a compartment with a membrane. But that could be some kind of intermediate level of self-assortment, of non-random self-assortment. And with that, like I just want to say that you can complain to all these people if you didn't like the talk. Mostly Jason for the ideas, because he was the one to start all this project with me. And you also have nice views of Bath and Marseille, which explain maybe why I'm so slow at writing manuscripts. And with that, I thank you all for your attention, and obviously the Society for inviting me for this talk.
We have time for questions. So do you get like also more just straight cheaters when there's these bigger groups, because they can free ride as well?
So in this framework, we don't, because we are in a situation, like that's the part I didn't show, but we are in a situation where cheaters are driven out of the population at first when they compete against altruists. And so if then you have a new green beard in the population, it replaces, even if you build a false beard, this false beard is going to be like a cheater to the altruist. And so if the cheater cannot invade the altruist, the false beard cannot invade its green beard. So that's not possible. The thing that is missing in this framework, and it relates a bit to your work, your previous work, is we don't have a way to build them. Like it's kind of answering the why more than the how you build the green beard, which was also an issue in the literature.